So welcome at the IPPN webinar organized by the Imaging Working Group. I think today we got an excellent topic presented by Waigo from the University of Tokyo. He's actually at the moment in Japan, where it's at this moment midnight, actually the next day already. My name is Rick van der Zedde. I'm in Wageningen University Research at the moment. And I would like to have just a few minutes of you just to show you who is currently involved in the IPPN Imaging Working Group. So it's Tony Pridmore from Nottingham, Hanno Schaar from Jurlief, Totosh from Edinburgh, me and Scott Chapman from Cyrone. We're organizing all kinds of things. So the webinar is one, but we're also working on workshops. The summer school, in the upcoming summer school in July, has registration. So from the 8th till the 12th of July, we can train you in imaging and with a focus, of course, on phenotyping. So if you want to know more, you can go to this website. So that's the IPPN website and look for the working group. So I won't spend much time on it because I would like to hear what Waigo is going to tell us. So the talk will be on easy plant phenotyping by using image processing and machine learning techniques. And another reminder, so if you have questions during the webinar or after the webinar, please put them into the chat window, because then I can ask those questions to Waigo in the end of the session. So I will now hand over the presenter role. 1 second. To Waigo, and then we can hear him talking and we can see his screen. So we cannot hear you yet. People can still hear me.
Can you hear me now?
Yes, I can hear you. We can all hear you. Yep.
Oh, that's great. Thank you very much, Rick.
You're welcome.
Yeah, I will start my presentation soon. Please wait a moment. Okay. So, thank you very much, Rick. And thank you again for this such opportunity to me to present our works and the thoughts to the plant phenotyping community. And it's really a great honor for me. And hello, everyone. I suppose I have to say good afternoon to the Europeans and good deep evening to the Asians and good morning to the American colleagues there. And today I would like to introduce how we are using the image processing and machine learning techniques to overcome the difficulties of the current plant phenotyping problems. Still have.
Yeah, so audio is now, your audio is now gone. I cannot hear you at the moment. Yeah, so I made you a presenter again. I'm not sure what you did. Yeah.
All right. Is that okay now?
Yeah, you're back again. Yeah.
Okay, great. So I just don't want to waste the time. So, I'm not sure if I was my voice was going to the first a couple of minutes, but I just go directly to the presentation I would like to give for today.
Please go ahead.
Yes. So First, please let me to take several minutes to introduce our team. So we are called University of Tokyo International Field Phenomics Research Lab, which is a new established lab at Japan. And as you can see over here, we are actually located at the Tokyo city, so which is surrounded by all the buildings inside the city, and we have quite good access to the farm we are working in. And as you can see over here, we are running a whole bunch of the both hardware and the algorithm developments for the plant phenotyping. And our topic is mainly focused on this three stuff, which is image-based phenotyping and field sensing techniques, and at the end of the algorithm, big data. As you can see here, we are quite international team, and we are developing those both hardware and the algorithm in order to help the breeders for accelerate their plant phenotyping. And for me, my research interest is mainly focused on the developing the image processing and machine learning based phenotyping solutions in different scale. Just for instance, from the cell to the whole plant community. So let's go directly to the processes of the image processing and machine learning based plant phenotyping. So what is it? Just like the other previous machine learning related webinar, the whole process will mainly included three parts. As you can see over here, the first part would be input the image, and the second part would be this big steps, which is more related on the image processing by using different strategies. And the last part is to output the phenotypic trends that you want. And I do believe that the only difference in between the traditional machine learning and the deep learning is that if you have to do this feature extraction and machine learning model selection for the whole processes. So as we can see here, if you run the deep neural networks, most probably you will just only need to do the image input and waiting for result output in short words. So let's just take an example from our set. Actually, this is a very old work of my supervisor, which is done in 1980s. So as you can see here, the images are collected by the camera, and it will be inputted into the computer. And the next thing what you want to do is to you can do the image pre-processing and extracting the features from the images and find out a best model that you want to have to extracting the trends, or you can just directly use the images, put it into a neural network to achieve the phenotyping purpose. Let's say in this case, it's to select the best soybean lines from a big amount of the candidates over here. So how are we going to make it easier? Actually, this is a very difficult question to answer. Luckily, in order to make it easy, our goal is very clear. As you can see over here, our goal is to, instead of the human eyes, we wanted to use the machine eyes to simplify the measurements. And there are a lot of components, as you can see over here. We have to take care, for example, the cost, the operation, the algorithms, and the library, and the tools, and so on. So I would like to shift to today's agenda now. So I will review 3 case studies of our team we did before, and I would like to propose an image challenge for field plant phenotyping to IPPN, and I would also like to introduce our new journal to all of you. So first, I would like to start with my first user case, which is focused on the vegetation segmentation under the field conditions, as this is the most of the image processing based phenotyping requires. So it requires to extract the region of interest. Let's say in this case is the vegetation area from the images taken in the field, and in order to use it for the future analysis. So what is the request and the challenges for this case study? Actually, it is straightforward. As you can see over here, under the changing light conditions, we want first to extract the region of interest, which is saying like pixel of the vegetation accurately, and also we don't want to spend a lot of time to use all the different models to suit for the different cases. We just want a single model to done this job very quickly. As you can see here on the graph, the different light conditions will affect the segmentation a lot. So what we do, actually, we have to thanks to the machine learning. We found that if you carefully choose the training data from the images, even we only use the color features at this case studies, we can still train a very good machine learning model, and we are happy to have this very good result for the segmentations. And we also provided a tool for the public to handle their own problems. However, after we run this job for a lot of cases, we just realized still, even though the user is using the tools, it still requires the users to choose the training data very carefully, which is somehow, as you can see over here, the users have to pick up the pixels very carefully that can cover all the light conditions from the database that he has. So we started to rethink about these problems, and thanks for the deep learning by the collaboration with the Inhang France team. Segmentation actually started to proposing a deep neural network that is still being tested for this moment, but we are actually expecting we will have a good enough global model to handle all the cases to segment the green pixels zone. As you can see over here, by using the existing already published data set, which is covering different variety of the crops, Simon trained deep neural network that can segment the green pixels, which looks very promising for handling all the different kind of cases over here. So I move to another case, which is the detecting of the flowering timing of the paddy rice. So why we want to detecting this flowering timing for the paddy rice? Because the frequent high temperature decreasing both quality and the quantity of the grains of the paddy rice, which is a very important crop for Asian populations. So the breeders started to try to develop the new varieties that can start make the paddy rice start flowering earlier, and actually they did it. It's very promising. However, in order to check the performance of such new developed varieties, they have to stay in the field from early morning and recording the flowering time, like what you can see over here, for several hours, which is very tough work. So here comes the request and the challenges for this study. So it's requiring to record the local weather information precisely for this experiment site, and it requires to observe the same plot continuously and stably, and it requires to detect those very small changes. As you can see over here, only 2 or 3 hours can show these small changes on the panics of the paddy rice, which we absolutely need the image processing and machine learning in advance. But before we start to run the image processing and machine learning, I think the problem is like we need to observe the same plot continuously and stably. And so for this case, we are using our own field monitoring system, which is developed by the collaborators in Naro, Japan. So this is called Field Solver, and it is open hardware and software sensor network. It can collect both images and weather information from the field very easily. If you do have interest on building your own sensor network, please visit this website to check the required hardware parts and techniques for building your own system. So after we have connected the images from the field stably, it comes to the problem of image processing and machine learning. So we do, as initial, we do really want to do detecting those small answers from the panickers, but same as it's very difficult for us. So alternatively, we started to change our idea to detecting the flowering pattern from the whole images. So for this study, we use the dense sampling strategy, and we are using the local feature descriptors, which is shift, and which will be applied in different scales and train a machine learning model. As you can see, the whole flowchart over here, as nothing changed with the basic processing as I introduced previously. So you have a part of training the images and extracting the features to train the machine learning model, and then you put the image to the model to try to extract the desired flowering patterns from the pictures. So this is a result that we got. It's quite well detected the patterns. Please be careful over here. We are not detecting the panickers, and we are also not detecting the answers from the panics. We're just detecting the flowering patterns from the pictures. So the number of these red blocks is not related with the number of the panics or flowering panics for this study. However, even though we're just detecting the flowering patterns from the pictures, we still can achieve the goals. So as you can see from these pictures, the x-axis is actually the time for each day. So you can see the flowering pattern very well, and from this graph, the breeders can actually grab the flowering time very precisely. So which is a good solution for them. However, it requires a lot of image processing works. So which we are thinking we have to transfer this to deep learning again. As you can see, before we do the deep learning, we have to make sure if the deep neural network really grabs the features that we want to emphasize the detection accuracy. And seems it works good for the case of the paddy flowering. So we started to build our model, deep neural network model. And again, I have to emphasize that for this case, we also do not build object detection network. What we do is actually we do the patch classification based on the sliding window techniques. So as you can see over here, every patches will be grabbed out where the window is sliding over the pictures, and the neural network will check if this patch belongs to the flowering panic or not. And then we can have the same results as the previous work we did, and we can detect in the flowering timing. And luckily, we found out another solution, solution for this work. Actually, we can estimating the key growth stage of the paddy rice, which is heading stage at this moment, as taking the cumulative number of the flowering panic patch. We can actually find out the flowering time for flowering stage, which is also the heading stage for the paddy rice for this case study. And then we are exactly find out that we can estimating the key growth stage of the paddy rice by just using this simple deep learning approaches. So as you can see over here, we are testing several different data sets and several different varieties and several different plant densities. It all works very good for the case of this study. Then I would like to move to the last case study. So this is a collaborate work with the Australian team. So the purpose of this case study is to detect and count the sorghum head number at the real breeding field. As you can see over here, the main problem is that if you don't have a good solution, so the breeders have to count more than 1,000 plots inside the field within a very short time to count the sorghum head. For this 1,000 plot is not an easy job for them. So what is the request and the challenge for this study? Since that's more than 1,000 plots, and that is a very huge breeding field. So we can't taking the pictures by hand, one by one, which is very difficult. So we started to think about by using the drones, which is also UAV. And the challenges is like we need to extract the macro plots from the UAV images, and we need to detect the various types of the sorghum head from the UAV images. As you can see over here, different lines just give the different expressions of the sorghum head over here, and we need to count the number of sorghum head per plot. And finally, we need to detect very small changes by using the image and the machine learning strategies. So before we start to doing the image processing, we need to have a very good pipeline to design the flight and the field experiment suit for this UAV case. And I think our previous webinar already introduced a very good solution for this part. So I just skip the details of this one. But if you want, you can visit this GitHub page that we are giving a wiki page for this whole bunch of the processes that can help you to start from selecting the drones and the sensors to do some basic phenotyping by yourself. And also, I have to emphasize that before the image processing and machine learning, we actually need a lot of basic pre-processing for these drone-based image solutions, as you can see over here. So we need to create also mosaic DSM, which is digital surface model, and the indices and point clouds from the raw images taken from the fields. And we need to do the plot segmentation, like over here, to make sure every picture is focusing on the plots, or we can say on the lines that we wanted to focus on. So besides of this process, we also have a tool that can easily help the breeders to view the images in time series order. So even if you don't have any techniques on image processing, you can still view visually and check what happens in the field, in your own field. So I have emphasized this part. I have put this red box on this part, which is saying like we need to extract the usable plot level images for the different cases. Why? For example, for the case of this head detection stuff, actually, if you are looking at the orthomosaic images, which is stretched by the original pictures, you can tell that the quality is losing a lot from this orthomosaic. So which is very difficult for the people who is willing to build their own machine learning and image processing pipelines for this kind of job. So we also proposed a work that can help you to do the plot segmentation in a semi-automatic way to provide the usable plot level images in the end. So my student will give a post presentation in the Phenome 2019, and you can also find out the detailed information in our GitHub page over here, and it will be online soon. So after we got this stable plot level images, we started to think about making the image processing and the machine learning models to achieve the final target, which is detecting the sorghum head and counting the number of the sorghum head per plots. So we proposed a two-step machine learning strategies, which is still a very typical machine learning and image processing solutions. So the first step is to extracting the candidates of the sorghum heads from the original images, and then we train those detected candidates with the corresponding number of age, and then we check all the candidate regions from original pictures, and then we give the predicted number of the heads from this picture. As you can see here, there are some small numbers which is not easy to see, but you can tell the candidate how many numbers are inside the candidate regions of the pictures. And also you can see the model is very robust to all the different varieties of the sorghum over here. No matter what color it is and no matter what shape it is, it will be still very clearly detected and counted. And we are happy to say that we have the solution to give the original quality of the plot images, and we can apply this model to the pictures, and we can have the results of the head counting at the end, and the result is quite acceptable at this moment. And as well, you can find out the detailed information from this GitHub page, and we are not only providing the detailed techniques of this case study, we also provide the data, the annotated data for this study too, so which can help you to quickly and easily apply this kind of work on your own data. And again, for this case, we also want to have a global model that can handle all the cases and do not require the breeders to think more on the image processing part. However, as you all know, if you want to train a deep neural network, the most important part at this moment is to labeling the data. So just put a small example over here. As you can see, this is only the central 10% of the data, image data we are showing previously. And inside this region, you have like more than 400 heads within this image, which means you have to do annotation 400 times. And even though you shrink it to the plot level, which is shown here, you still have more than 100. So please imagine how you do this big, huge label intensity work for labeling all of those pictures. You may want to say that, yeah, I can use a crowdsourcing, or I can ask someone to help me, but how you can make sure the quality of labeling is good enough to train the machine learning models. So in order to solve these problems, we are not fully solve these problems, but we have some thoughts on it. So we proposed a semi-automatic way to solve this problem, which we called a false negative and false positive relearning strategy. So basically, we started to build this web tool, which is available for the image annotation, and we do not do a lot of the annotation. So we just do like several pictures very carefully, and then we'll start to run the model based deep learning model based on these labeled images, like you see over here, and to train the model once, and to make the prediction for all the left-handed data sets, and then feed it back. So feed this predicted data back to this web tool again, and to make the human verify to check if the model missed some part or not. If it missed some part, add it. If it makes the wrong predictions, just correct it. So it's inspired by the conception of the active learning. It's still not fully automatically, but even though it's semi-automatically, we confirm that it actually saves 5 times speed of the compare with the traditional labeling work based on this data set. So finally, we had by rerunning this kind of process, we have a good amount of the annotated data set, and then we have a good deep neural network to be trained, and we tested it on the different varieties and under the different net conditions, as you can see over here, and the result is quite good. And we're going to also publish this model to the public soon after this paper to be accepted sooner in the future. So this is all the 3 case studies that I want to share for today's presentation, and I would like to go to the conclusion and the future plans for our teams. So I have to say, to make the plant phenotyping easier, we all believe that image processing and machine learning is a very powerful tool, but we still have to do a lot of work in order to make this image processing and machine learning easier than before. For example, as I presented previously, the data annotation is still a very tough task at this moment. And also, I think we are not only just focusing on the 2D images, we are also like to starting our analyzing on the three-dimensional images soon. So there is just a small example that we did last year. So we do the 3D segmentation based on the point cloud that is collected by the plant eye, which is LiDAR scanner. And then we would like to do the 3-dimensional growth tracking for each leaf in the future. And also, I think in order to overcome the first biggest problem of the machine learning and the image processing at this moment, we have to focus on the data annotation at this moment, so that we need more scientists to join the community at this moment. So we started to propose this image challenge for field plant typing to Rick several months ago to the IPPN, and we are thinking if we can provide our already annotated high quality data to the public, and we could start to ask those questions to the public, which obviously the first question would be, can you increase the accuracy of for example, the detections? And also, we do not want to spend too much time on the training data collections, which is annotation. So we would like to ask these questions like, if you give an image, can we get to the most needed target to be labeled and left the others to be labeled automatically? So it's a little bit tricky, but the key conception is like we want to save the labels to just focus on the bounding box that are really needed to be labeled. And also, giving a data set, for example, if you have 1,000 images, can we tell which are the best images need to be annotated? Not all of them, just a part of them is good enough for, based on our previous research, but we have to make sure which one would be the best to annotate it. So we already have a couple of the collaborators from all of the 4 countries at this moment, and we are still call for the collaborators. So please feel free to contact me if you want to join this kind of challenge. And at last, I would like to take this opportunity to introduce our new journal, Plant Phenomics, which is a science partner journal that are supported by Nanjing Agricultural University in China. And it's just launched 2 days before, and we are called for the papers at this moment. So you can simply scan this QR code to get more detailed information of this journal. And on the behavior of our editors-in-chief from Japan, France, and US, we are officially started to call for the papers from all of you. So please feel free to take a look at the website of this journal and get the detailed information from it. All right, I think this is all for my today's presentation, and I'm looking for the questions. And you have more questions, you can scan this QR code to send an email to me directly. Thank you very much.
Yeah, thank you. Okay, excellent topics. So we do have already some questions. So you referred in the last section to the image challenge. And I also would like to share with you the publication that has come out by the 2 people from the working group, by Sotosh and Hanno Schar, is sharing the right data right.
Yes, yes.
Their image data set for the leaf counting challenge in Arabidopsis and tobacco was downloaded and used extremely often also outside our own phenotyping community. So I added the GUI in the chat window. So, but anyway, let's go to the questions. Got several questions. I have some questions, but I'm gonna ask your questions first. So okay. Monica asked around slide 22, but I think it's a general question. Is there a library for feature extraction that you use?
Oh, okay. So actually, this is a very difficult question to answer because there's a lot of libraries that can help you to do the feature extractions. So basically, if you are new for the image processing, I would like to recommend MATLAB with image processing toolbox, and you will find a lot of useful functions to do this kind of feature extractions. And if you are familiar with Python, I would like to introduce you the OpenCV and yeah, Python and OpenCV. And there's also another library which is Plant CV, which is also very good to take a look at it. And there's a lot. So maybe you can just send me an email. I can give you a couple of links of this those libraries.
Yeah. And do you have specific algorithms that you use or that you can recommend? The second question from Monica.
Oh, thank you very much for your questions. And this is a very tough question. Actually, for the different for the different purpose, you need the different algorithms, obviously. So there's not a specific algorithm I can recommend at this moment. But I mean, it's all depends on what kind of purpose or what kind of final target you are looking for from your images. I'm sorry, I can't just easily answer this question at this moment.
Okay. And I've got a question actually in the meantime. Like at slide 22, actually, you show differences between varieties.
Yeah.
And can you also explain if there are breeders involved that are using the data and how they appreciate your automated analysis?
Okay. You mean this one, right? Or maybe this one.
Yeah.
Yeah. Yeah. I mean, this is actually I got a thanks message from a breeder who is really working in the field and who is really doing the countings in the field. So basically, what he's doing, like he cannot counting all this 1,000 plus plots. So he just take some samples to counting the number of the heads, and obviously it's not achieved his goal. So by this kind of approach, he can have his answer for whole plots, which is we can call it like full sampling strategy for his experiment, which is really helpful for the breeders at this moment. And we are going to provide more integrated tool to the breeders to help them do not run the code on their site, which I'm presented just now. We are transfer these techniques to the deep learning to make everything simple for them.
Yeah, because that in the near future, breeders with some positive attitude toward automation simply use your methods, and you don't have to interfere anymore. So they just fly over their fields with the drones. They run your sorghum head counting algorithm and run that into their, like you say, breeding pipeline analysis.
Yes, we're going to provide a real-time counting board, which is integrated with UAV this year. So I probably have another chance to show off you sooner within this year.
Okay. Because we are also aiming to offer breeding colleagues from plant breeding those tools.
Okay, that's great.
Question from my colleague, actually, Gerrit Polder. Do you have any experience with other image modalities than RGB, for instance, multi or hyperspectral imaging?
Oh, yes. At this moment, thank you for the question. And I have to say, at this moment, we do also do the same processing for the multi-spectral images, but we don't have any experience at this moment for the hyperspectral images. So, I mean, the whole process of this pipeline is okay with multispectral images too. So we are running the same stuff on it at this moment. And we also have the indices, which I'm mentioning over here, but I don't show the examples of the multispectral images since of the time limitations. But yeah, we do the multispectral stuff, but we don't have the hyperspectral at this moment. So we would like to have collaborations with people who is running on this hyperspectral images analyzing too.
Mm-hmm. Okay, cool. Another question from viewer 220. Then the number says something already.
Okay.
Viewer 20 asks, what is your turnaround time from raw UAS images to images of individual plots labeled by row and range number?
Okay, thank you. So what is the turnaround time for raw UAV images to images of individual plot labeled by? Okay, this is a big question. I'm sorry, I can't answer it directly, but I have to say, like, in the case of this study, so the raw images is around 200, and we are running pixel 4D to get this auto mosaics and those point clouds. It will take around 3 hours in my machine, but my machine is a little bit strong, which have 2 graphic cards and a big memory and 2 CPU on it. So it's strongly depends on the spectrum of your computer. And then from the auto mosaic to the usable plot level images, which is raw images, it's not taking too much time. I didn't count the time, but it's just acceptable time for this process. Just several minutes from one auto image to several plot images. Is that answer your question?
I hope so. So is it all fully automated pipeline on this slide with the basic pre-processing, or is there quite some manual adjustment needed?
Oh, yes. At this moment, from raw images to the auto mosaics, since we are using a commercial software, which is Pixel 4D, so it's we are running some batch processing on it, but it's still not automatically done. And from auto mosaic to the basic analyzing of the plots, it's almost automatic at this moment. But you still have to manually run this process by using the commercial software from my side. So sorry about that.
Another question from 220. Are you analyzing auto images of plots or raw images?
Oh, very good question. Yeah, I do both. Why I do both? So it also strongly depends on what kind of final target you want. For example, if you just want the canopy height, or if you just want a growth curve that can be got from the canopy coverage, it's all right to using the auto mosaic of the plots. But if you want to do some more processing, I mean, like this, detecting the heads, which needs more detailed information from the images to developing the algorithms, you probably need to analyzing the raw image. So to simplify my answer, if I do the head detection of the sorghum, I will use the raw images, not this big mass auto mosaic images. But if I just calculating the growth curves, I will run the orthomosaic images because it's saving a lot of time. Is that okay?
Yeah. So I had a question specifically about this question, about this image that you have now in front of you.
Yeah.
Is this the average quality of these orthomosaic images?
Yes, most probably.
example?
Yeah.
Okay.
Yeah, because this actually I have to go back to the pipeline again. So from here to here, from the raw images to the orthomosaic, which they need a very good feature point detecting and feature point matching to run this we call it structure from motion stuffs. But most of the time the structure of the plants are very similar, so which is very difficult for it to have a good feature point matching. And in other side, the plants are not stay there steadily, so you can't make sure every time if you take the flight, the plants are not moving neither by themselves or by the effect of the wind. So you will never have a perfect picture, original picture that makes the plant stay there perfectly. So always it will give like this kind of quality loss if you run the orthomosaic stitching stuff.
Okay. Another question: did you notice also that the drone itself was affecting the movement of the plants?
Oh, if you fly in a low altitude, yes.
Okay.
But if you fly like higher, there's no problems.
Okay. So I have one last question, but if somebody else has a question, please put them in the chat window. I was actually quite fond of this relearning tool that is web-based.
Yep.
You can like simply have the human in the loop to give feedback on like the first version of your deep learning network.
Yeah.
Is this going to be also generalized for the public, or are you going to put that somewhere so others can also use the same approach?
Oh, this is a very good question. Actually, we are facing some budget problems, which is also related with how to make this kind of work easier. Because at this moment we are using AWS, which if every time if you load the images, they're going to charge us. So we just cannot put it online for public use. But we do willing to share the source code in the near future so that you can just build your own server in your local conditions that you can run this process on your own set. But it's not easier for us to make it public, online public, really.
Now I can imagine that the source code is more efficient than the tool itself, because every image set has its own specifics.
Yeah, right.
Okay, cool. So if there are no further questions from the audience, then I would like to thank you again for actually really inspiring talk. So I will definitely continue talking on this topic, but then offline. I'm sorry.
Yep.
There's a question.
Okay.
Can you recommend any conferences or events where we can see more of your work?
Oh, thank you very much. Yeah, I have to put the website of our laboratory, but I forgot it. So please, if you can send an email to me, I can share more information to you. So this is a barcode and my email address over here. And also, if you have some connections with IPPN ImageNet work group, they must recommend a lot of very useful conference for all of the communities.
Yeah. So you're going to go to the Phenom in Tucson?
One of my students will go there, so you can see one of our work over there.
And I missed one question, but I skipped past it. The magazine, the journal. Somebody, I find the question. Somebody asked like, what journal is it? So just can you put it back onto the screen?
Yeah.
The Phenomics journal.
Journal?
Yeah, it was slide 36, I think.
Okay. Journal. This one?
Yeah.
Yeah. Call for the papers.
Yeah. Okay. So that's
Thank you very much.
for today, everybody. And, you were a wonderful audience, and we never had that many participants as we had today. At the peak moment, we had about 124 people signing in.
Oh, that's great.
That's an enormous amount of people. Yeah. So clear sign that at least the community is growing, and imaging in phenotyping is a hot topic. So thank you for the talk, and have
Thank
a
you
nice
very much.
good night to you. Actually,
Yeah, thank you all for you.
Okay.
Good night.
Thank you.
Okay. Bye. Stop. Stop.